Alrighty, let's begin Unit 2 on cell structure and function. In this unit, we're going to explore cells as a unit of life and how do cells contribute to the organization of life and provide the environment in which organelles can function. Cells and cellular organelles, they have membranes that enable them to establish and maintain an internal environment and control exchanges with the external environment. This maintenance of internal and external cellular environments is what we call homeostasis, a word I'm sure AP biology students are familiar with. An understanding of the principles of cellular homeostasis is important to understand the material in this following unit. We're also going to utilize some skills by being able to explain relationships between the cell structure and the cell function, the function of the organelles, and all the cellular components on a subcellular and cellular level. That was quite a mouthful. But you should become proficient in describing data, making calculations, and analyzing different types of data through this unit, as you've been introduced to it from Unit 1. All right, cellular structure and function, and subcellular components and compartmentalization. So let's talk about an organelle. In this video, we're going to talk about organelles, more specifically, energy organelles. So endosymbiosis, the endosymbiont theory, is a theory that explains the similarities that mitochondria and chloroplast have in a prokaryote. This theory states that in early eukaryotic cells, they engulfed a prokaryotic cell, and that prokaryotic cell became what's called the endosymbiont, or a cell that lives within another cell, and they became one functioning organism. We do have evidence of endosymbiont theory, and that is a double membrane. So there's a membrane on the exterior of a cell, and there's also a membrane that surrounds these energy organelles. Ribosomes are also other evidence of this theory, and also the circular DNA that we find inside of the mitochondria and plasmids of prokaryotes. And, really the idea that just these energy organelles are pretty much capable of functioning on their own is really enough to explain this theory. Mitochondria, that is the site of cellular respiration. So that's the structure of the double membrane. The outer membrane is very smooth, and the inner membrane, called the cristae, it has these folds in it. So it divides the mitochondria into two internal compartments. And the reason for it is increasing surface area. So the little squiggly part that you see increases the internal surface area inside of the mitochondria. Mitochondria, the inner membrane is the space between the inner and outer membrane. So the matrix is enclosed by the inner membrane. The squiggly part that you see inside of mitochondria, that's called the mitochondrial matrix. That's basically the Krebs cycle. That's where the Krebs cycle happens. There's enzymes that are there, and they catalyze cellular respiration and produce adenosine triphosphate, ATP, that actually has its own DNA inside of the mitochondria. And they have ribosomes that are specific to just mitochondria. So the number of mitochondria in a cell will correlate with its metabolic activity. So cells that are going to have a higher metabolic activity, they're going to need more energy. They're going to have more mitochondria. These are cells like muscle cells where there's a lot of movement and contraction. The chloroplast, fascinating and specialized on its own. This is a really specialized organelle in that it is part of what makes a photosynthetic organism. It's where photosynthesis happens. It contains the green pigment, chlorophyll, and inside of its double membrane are thalkaloid sacs. Those are membranous sacs that can organize into sacs called grana. And that's where the light-dependent reactions occur, is inside of the grana. So one of the things that I will use is when you bring a big stack of pancakes, it's a grand stack of pancakes, a grana stack of pancakes. stroma is the fluid that's around those stacks, and that's the location for the Calvin cycle. It contains chloroplastome DNA, specific ribosomes unique to the stroma, and then really specialized enzymes that assist in the Calvin cycle. The cytoskeleton. The cytoskeleton is a network of fibers throughout the cytoplasm that give it structure and support. This is especially important for animal cells because they need some sort of mechanical support without a cell wall. So the cytoskeleton fibers, they anchor organelles and they allow for movement of those vesicles in and out or just around the cell as a whole. And so this movement occurs when the cytoskeleton interacts with motor proteins. There's three main types of fibers in the cytoskeleton. They're microtubules, microfilaments, and then these intermediate filaments. And even though they look similar in illustrations, they do have different functions. So microtubules are hollow-like rod structures. They're made of protein. It's a protein called tubulin. We get real creative here. And they grow from the centrosome. They're going to assist in that microtubule assembly. Microtubules are fascinating in that they just appear. They mainly serve as a structural support. So if you think about it like tracks that support a train, we'll just say. So for the movement of these organelles that are interacting with motor proteins. So it's like a train track kind of support system for other motor proteins, but it assists in the separation of chromosomes during cell division. That's what they're famous for. They just appear during mitosis, and they pull the DNA chromosomes apart. But they've got some other function too. You know, they're part of the motility of the cell, so they're also contractual structural fibers that are inside of the cilia, which are the hair-like structures of cells, and flagella, that whip tail-like structure. Microfilaments are very thin, solid rods. They're made of a protein actin, and they have a different function. They maintain the cell shape by bearing tension that the cell is experiencing. They assist in muscle contractions, and cell motility is usually where students learn about them. But actin, that protein, works with another protein called myosin. And when these two proteins work together, we get a contraction. It also assists in the division of animal cells, and this contractile ring that it's part of, that cleavage furrow that forms when cell division is occurring. It's easy to get microtubules and microfilaments mixed up. I mean, they do perform similar yet different functions, but really the difference is the proteins that they're made out of. Intermediate filaments are fibrous proteins. They're made up of a variety of subunits. We're not going to learn specifics, but they're a permanent structure of elements in the cell. They don't just appear at special times during the cell cycle. They function to maintain cell shape, and they anchor the nucleus and other organelles inside of the cytoplasm. They are most known for forming the nuclear lamina. That is the space that's in between the chromatin and the inner nuclear membrane, a space that essentially holds all of the DNA that when you can't see it, when the cell's not going through a type of division or anything where it hasn't coiled its DNA. And so it lines the internal part of the nuclear envelope and provides that anchoring system and shape. You can be asked to identify an organelle that is responsible for something or describe steps that you would take in a procedure to determine if it was that organelle. So let's try this practice FRQ